I formally, thank the CGC, all the members, to provide me this opportunity to share my work. Let me tell you something what I have been doing in India and then in my subsequent postdoc. So I had, earned my PhD on Red Junglefowl genetics. So I have been studying Red Junglefowl and the chicken in India since 2007. So approximately, I have spent more than 10 years in understanding the chicken, their genetics, genomics. So while working in India, I had worked more on the genetic aspects, and then I had addressed the genetic introgression of Red Junglefowl with domestic chicken. And that was a kind of my PhD work. And while working on the genetic hybridization on swamping of junglefowl, I got interest to work in the chicken domestication because we were collecting sample all across the country. So wherever we had sample of junglefowl, we collected domestic chicken samples and the country and village chickens. And in my subsequent association with the Kunming Institute of Zoology in Chinese Academy of Sciences in China, so there I have taken up a bigger aspect where we have addressed the origin and chicken domestication. So there we have sequenced approximately more than 800 genomes, and this is like on 10x coverage we have sequenced, and then we try to understand what are the center of domestication and which species of Gallus that has contributed to the domestication. So I would be sharing the key learnings, and I would say it's not the end. We are still in the chicken genomics, and we are trying to understand the underlying mechanism of selection, diversification of chicken into different breeds, and we are trying to understand those genomic regions which are under selections. And there are certain breeds which is very fascinating. Like one we have in India is called Kadaknath, that is complete black in color. Even the meat of the Kadaknath is complete black. So we are trying to explore the underlying mechanism and the diversification of chicken into the different breeds. So while carrying forward my work from PhD to a postdoc, and subsequently I work as an associate professor in Chinese Academy of Sciences. So there I had opportunity to coordinate and between 15 different countries, and about 7 TB data we have generated for this. So now let me jump to the slides straightforward, so that we'll have a straightforward understanding what we have done. So let me share the slide. So chicken domestication is always been in controversy, and it is not so straightforward like many other livestock. And time to time, the scientists has debated and proposed different thoughts about the domestication. And so I chose the title saying is a multifaceted and complex history, because it's not like it's a monophyletic, it's a polyphyletic origin of chicken, and the center where the chicken has been domesticated has multi layers to explore. So I would be touching up and addressing these questions: the genetic endangerment of Red Junglefowl. So whether it is real or is just a theoretical. People say the swamping or the genetic introgression from domestic to wild is just a theoretical phenomenon, or whether it is real. So this I have explored, sampling from India, and then origin of chicken, whether it's a monophyletic or polyphyletic. And we know the Red Junglefowl, species, the Gallus is a polytypic species where there are 5 subspecies in the species, the Junglefowl. So which subspecies of Red Junglefowl has contributed to chicken domestication, and whether it is a hybrid origin of chicken, because there are a few scientists from Europe. They also propose that probably chicken domestication is of hybrid in nature. So this work, what I'm going to present today, I had worked in Wildlife Institute of India, being in India in Dehradun, and then Kunming Institute of Zoology. So these two institutes I have been associated while exploring these questions. And my friend Nishant, he is also from the same institute, the Wildlife Institute of India, so where he has studied his master's. So I had studied my doctorate from the same institute. So let me introduce the genus Gallus. As we know, it has a 4 species of in the genus Gallus. The grey junglefowl is called Gallus sonerati. The Ceylon junglefowl, endemic to the Sri Lanka only, is called Gallus lephiti. The green junglefowl, Gallus varius, and the red junglefowl. Sorry. Red Junglefowl, which is again a polytypic species and has a 5 subspecies within it. So the Gallus murgi is found in the Indian subcontinent, and some part of the northeast India we have a Gallus species in India. However, it has a distribution in the southwest Yunnan, Myanmar, Thailand, and Peninsular Malaysia and Sumatra. Gallus jeboli is found in northern Vietnam, southeastern Yunnan, and Hainan. Gallus is found in Cambodia, central and south Vietnam, and the south Laos and the Thailand. And Gallus, which is endemic to Sumatra, Java, and Bali Islands. So this is the about the entire the genus Gallus and the 5 subspecies present in the Red Junglefowl. So this image we have taken from the museum from UK only. It's a John Carter picture which shows a little phenotypic differences between the 5 subspecies of Red Junglefowl. So, however, people has time to time reported even hybridization between these 5 subspecies, because at many places they have an overlapping distribution, and these subspecies also reported to hybridize with their domestic counterpart, the modern day chicken. So, with this background, I also would like to demonstrate these photographs that I have taken from India. And these shows the close interaction of the Red Junglefowl, the Gallus murgi, with the domestic chickens, the male and the female. They found the same flock. So I was trying to trap these birds during my PhD there. So we have found these guys, mixed with the domestic chicken on the fringes of the forested areas. And people also collect, Red Junglefowl eggs from the forested areas, incubate them with their domestic hen as a surrogate mother, and there are chances of, hybridization between these two. So we know there are several hundreds of, breeds of domestic hen all across the world, and the what we believe that Red Junglefowl is the sole ancestor, and it has a application in several ways. They have influenced the mankind, as multi-dollar poultry industry depends on it, and people are using different breeds of domestic hen for cockfighting. Is used as a game bird. Is the first agriculture bird and the whose genome sequence. So it's a role model for the embryological studies and several other research studies. The Japanese scientists, they made some unconfirmed in silico experiment where they have given the doses of the H5N1 avian flu virus, and they found this Red Junglefowl is 10 times more resistant. So there are something in the genome of the Red Junglefowl supposed to be the wild ancestor. So his genome is important, and wherever we have to address certain immunogenetic response in the domestic chicken, and we have to diversify different breeds, we have to go back to the wild progenitor. So these guys, the Peterson and Brisbane, probably the first scientists who has reported the hybridization, and they have proposed several traits like eclipse plumage in Junglefowl in the post-breeding season, the slendrous grayish color lack of Red Junglefowl, and the female does not have any comb or rudimentary comb in their lifespan. So these are the characters they have proposed. But late in 2007, they said these characters, they get reappeared after backcrossing. So these characters may not be used. The phenotypic characters may not be used for assessment the purity of Red Junglefowl. So we did the survey for 2 years in the entire India, and then we mapped the distribution and also the diversity pattern, genetic diversity pattern of the different population of India. So so we found that it is present in 21 states and in 25 districts. And we use this foothold snare traps to capture the Junglefowl, and wherever we capture Junglefowl, we have withdrawn the 1 ml blood sample and for genetic analysis. So while doing landscape genetics, so it's like a Bayesian cluster analysis, and we reported at least there are 4 distinct genetic clusters. So if we consider the landscape features along with the genetic assignment, the population assignment, we found there are at least 4 distinct population of Junglefowl. So if you see this map, there are 4 distinct clusters. So we say like all across the country there are at least 4 distinct populations, and then we have because I was working on the genetic hybridization, so we have also traced the birds in captivity, and we found there are certain birds in captivity which were hybrids. The green one is the junglefowl that we have sampled from different zoos, and the red one is the domestic chicken. So you see, in between there are few individuals which were hybrids. So we have marked these birds with the ring number. So ring number. So we have identified these birds after the genetic analysis over, and then we have submitted report to the zoo authorities that these birds are hybrids and can be removed. So we have also done, this is the haplotype network based on the mitogenome. So we found there are certain haplotypes which were common between the junglefowl and the domestic chicken. So the hybridization or the introgression, we say, is real, it's not theoretical, which has been proposed earlier by several scientists. So this is kind of a result I have found during my PhD. Then now coming to the origin and the evolutionary history of chicken domestication. This probably the classical, the first paper that talk about the monophyletic origin of modern day chicken. So these guys from Japan, they have analyzed 21 birds, of which there are 4 species of Gallus and 3 subspecies of Gallus. So they have a Gallus spadiceus and Gallus bankiva. So this says the author reported that the Gallus is the real ancestor of all modern day chickens, though they have less samples and they have not sampled all the 5 subspecies of red junglefowl, but still they said the Gallus subspecies of red junglefowl is the ancestor of modern day chicken. So the Gallus from the ancestor to the domesticus, Gallus domesticus. And they also reported that the 3 subspecies of the red junglefowl are non-discriminable based on the genetic analysis, and their study was based on a small region of mitochondrial genome. Then this study was published by the European scientists, and they say and supported that domestic chicken origin is probably a hybrid origin, and where they reported that domestic chicken has a yellow skin, and the red junglefowl has the white skin. So yellow skin marker or the gene is contributed by the gray junglefowl. So this is the first study that talk about the hybrid origin of domestic chicken. So yellow skin is because of a gene called beta carotenoid. So because of this gene, the skin of the domestic chicken become a little yellowish. However, if you see, or if you have hold the junglefowl, you will see their skin is a little more transparent or whitish in color. Then this study was then published by the Indian scientists, and this talk about the multiple domestication of the modern day chicken. So it's not like only the Gallus murgi that has contributed, which was proposed by the Japanese scientists, but this is the murgi, the subspecies Gallus and the spadiceus. These all 3 subspecies have contributed to the chicken domestication, and it has been done on multiple places. So they supported the hypothesis of multiple domestication of modern day chicken. And this study also reported that red junglefowl is not being hybridized, which has been reported earlier. So they said red junglefowl is relatively pure. There's not that much hybridization happened. So this is what was the background when we started this project, or I have contributed to this project. So this project was was sort of a mission where the 15 countries have participated, and I have contributed from India part only. So all the samples that I have collected during my PhD days, those we have sequenced for the entire genome analysis, and all sequences was carried out in India. So you'll see the sample size here. We have sequenced approximately 863 genome, and see the coverage of these samples. So we have sampled the all 4 species of genus Gallus and all 5 subspecies of red junglefowl. And all the samples that we sequenced was at least 10x coverage, so that we have a compatibility, and then we could retrieve the enough variants, or we say the SNPs, so that we can do the subsequent analysis like assignment, structure assignments, and mixture analysis. And approximately 7 TB genomic data we have generated for this project. So this project took almost 5 years to complete, and in 2020 we came out with this publication, Cell Research. So if you see here, this is the first ever report that talk about or shows that 5 subspecies of red junglefowl are can be resolved based on their genome. So earlier workers, they said they have a overlapping genomes, and there are intermixing between the subspecies of red junglefowl. This is the first study, and based on the entire genome, whole genome analysis, we could classify the 5 subspecies of the red junglefowl separately. And you see the Gallus bankiva form the basal clade of all the other 4 species of red junglefowl. So Gallus bankiva is little, the older one, or the most recent ancestor one of all the other 4 subspecies of red junglefowl. And this was based on the autosomal variants, this phylogenetic tree, and this tree was based on the entire mitogenome, which is approximately 16 kb in size. So here, one thing is very surprising. If you see the geographical distribution of these subspecies of red junglefowl, the Gallus jabberli is on the peripheral part, or on the periphery of the species. So it is distributed after the jabberli. But if you see in the phylogenetic tree, so jabberli is becoming intermediate population between the Mugi and the species. So this is something surprising, and this pattern was constant between the phylogenetic tree based on the autosomal variants and the whole mitogenome. So this was little surprising, and still we don't have an answer for this, why this jabberli made an intermediate population between the Mugi and species on phylogenetic analysis. However, it is on the one side of the distribution. So probably this might be, we can assume this is a present distribution what we know today, but we don't know what happened before the LGM, or before the Holocene period. So probably this might be because either they have the large dispersal or the distribution up to the Mugi. So there are something which we could not catch or we could not answer with the present data set. But yes, we could classify all the 5 subspecies of the red junglefowl separately. And this is again a admixture analysis, and you see this admixture, the Bayesian admixture analysis, and this is a non-Bayesian PCA. So both corroborated, and you see all the 5 subspecies of the red junglefowl, they are, resolved. They made a distinct cluster with a little admixing. So we have a large sample of the Gallus Mugi, because these I sampled mostly from India. So approximately 148 samples we have sequenced for this Gallus Mugi. So we have a good data of Mugi, but you see the other populations also, have made a distinct clusters, and this based on the PCA also, these all form 5 distinct clusters. So we can say based on the whole genome analysis, we can distinguish or discriminate the all 5 subspecies of red junglefowl. So again, admixture analysis with inclusion of all the domestic chicken samples that we have collected from India and the other part of the world. Also have samples from Europe and some part of the South Asia, North China, and Western Asia, South China, and Southeastern Asia. So if you see, the Gallus species, they found closer to the domestic chicken population. And here again, the 5 subspecies of red junglefowl are classified into the different clusters, and this is how the other breeds are classified in the admixture analysis. So while putting all the domestic chicken samples collected from widespread range along with the 5 subspecies of the red junglefowl, we found that the Gallus species form the basal clade to all the domestic chicken breeds. So we can say the Gallus species. Sorry, I don't know what is happening. Why it is changing again? So Gallus species here form the basal clade to all the domestic chicken, all the breeds of domestic chicken collected from different ranges. And here the Gallus bankiva again form the basal to all the 5 subspecies of red junglefowl. So this is the primitive one. So then we wanted to understand the split time when this divergence would have happened, because now up to now we know this Gallus species would have been the ancestral stock of the from where the domestic chicken originated. So we try to understand the pattern of the divergence. So this is the MSMC plot based on the relative cross coalescent approach. And so here, if you see the red one is the Gallus mugi and the chicken. So divergence time is approximately, if you see, is like 9,000 to 10,000 years. So probably the domestic chicken diverged from the Gallus species in last not more than 10,000 years. And here also we try to understand the genetic ancestry with of domestic chickens. It's like the gene flow analysis of domestic chicken breeds with the 5 subspecies of the Red Junglefowl. So you see they have 3 statistics estimates show the highest for the Gallus species. So this confirms and further validate that Gallus species is the one which has the highest gene flow with all the domestic chicken populations collected from various ranges. So this is what we have done in to understand the evolutionary history of the chicken domestication. So is coming to summarizing the results. We know the chicken genome is a complex, and the event like introgression, selections, diversifications made this really multifaceted, because it's like a reverse and the forward introgression. And now we know if a population which we have domesticated, if it go for the reverse introgations, and even the exchange of the birds between different regions and countries, then make multifaceted to understand the domestication. If it is say one-way event and one directional event, then one can easily explore and understand. But it has have a mixed ancestry where even there are evidences of introgression between the subspecies of Red Junglefowl and also with the domestic chicken. So see the reverse introgression make it more complex to understand the evolutionary pattern or the origin of domestic chicken. So we could classify the 5 subspecies of the Gallus. They can be resolved at genomic level, and as I mentioned that we really could not understand that why Gallus jaboli clustered in between the Gallus mugi and species in phylogenetic analysis, while it has a different geographical distribution. So the split time between chicken and the Gallus species is approximately 9,500 years, that coinciding with the period of Holocene, and this is the time where even I believe the wolf also domesticated during the same time. And most developmental activities, because I believe the after the the algae that human drive and the civilization happened, and this actually if you compare this timing, the 9,500 years in archaeological data, I believe this is much later. The archaeological chickens are archaeologically visible much later than this split time 9,500 years. And we found this Gallus bankiva is the most divergent subspecies of the 5 subspecies of Red Junglefowl, and has a time to the most recent common ancestor with the other RJ subspecies. And it's about 500,000 years ago this got diverged from the. So the divergence of the 5 subspecies of Red Junglefowl is really older than the chicken domestication. So first they have diversified into the different subspecies, and then we had this chicken domestication. Yeah. So considering the this current distribution of the Gallus species, we can conclude that chickens are domesticated somewhere in the southwestern China, and it's not originated in India as thought previously, that Indus Valley or the nearby region is the center of domestication. So we have this study provided different results, and we support that chicken was domesticated somewhere in the southwestern China, probably in the Yunnan and the Sichuan provinces, in the western Thailand and Myanmar. And after the chicken got domesticated, then they would have been transported across Asia, and then they might have been interbred between the different subspecies of red junglefowl, and even with various species of junglefowl, the Gallus. So yeah, this is what I wanted to discuss. Yeah, thank you so much.